is quite familiar. So I'll start with my presentation. Where is my presentation? Here. So I'm here to talk about advances in neutralization assays for viral vaccines. So I'll take you through all the aspects of neutralization assays, but let us go a little into the history to see what has happened. So this is just for our basic understanding how the modern day mobile phones are there today, the Android or the iOS based. While in 1876, when Alexander Graham Bell invented the phone, it had only one feature. It could only transmit speech electrically. Whereas if you look at today's mobiles, it has all those features, like it has operating systems, cameras, GPS, modems, high-speed data modems, touchscreen. So why I'm showing this is, like, all these inventions were not meant to be put into the mobile phone. All these inventions happened over the time in the field of telecommunications and electronics. So similarly, when we come back to our field of vaccines, the neutralization assay didn't just appear all of a sudden. In 1796, when Edward Jenner first demonstrated how a cowpox vaccine is effective against preventing smallpox infection, that time nobody knew what viruses are. Only bacteria were known, but there was no idea of what viruses are. The first virus was discovered, reported in 1892. That was the tobacco mosaic virus. That is like almost 100 years later from the smallpox vaccine development. Then the important one I'll just cover for the sake of time. Renato Dalbecco in 1951, he first developed the plaque assay, and he showed that viruses can be quantified by in vitro culture. And he has several other contributions in the field of science. He got Nobel for reporting the reverse transcriptase and the role of oncogenes. And he also is very well known in the field of buffers, all the Dalbecco buffers which we use. Then in 1967, the pseudoviruses were reported, and finally in 1971, the ELISA was invented, where, like, from there the field just got widened and broadened, and then we have, like, tools by which we can measure the antibodies or cytokines and whatnot. So the neutralization assays are like a mix of all these knowledge which has happened for last 200 years. So when we develop a vaccine, there are like few very common tests. I'm talking only about the virus specific part, not talking about sterility and other whole list of testings. So there is a virus identity that we can do by several, say, real-time PCR or by some monoclonal antibody detection. Then virus titration is there, and antigen content is there. Similarly, in drug product also similar tests are there. But when we go to clinical or preclinical development, there are like two main aspects. One is the viremia, that is mostly for the live vaccines, because the live attenuated viruses will replicate in the body. So we have to check how much the virus is replicating in the body. The second one is neutralization. Now that is applicable for all type of vaccines, because once the antibodies are generated, we have to show that these antibodies are effective and able to neutralize the wild type virus. So this is a very basic schematic, the plaque assay developed by Renato Dalbecco. So what we do is we just put a heavy overlay using methyl cellulose or agar on the cells infected with virus, so they don't escape into the virus cell culture supernatant and spread across in the cell culture. So they just restrict their growth and they form plaques. So this was a continuation of the bacterial colony formation. So Dalbecco modified that method, and then when the in vitro cell culture system was established during 1950s, this method was developed. So there have been some improvisations in doing the plaque assay. The first one, neutral red stain was used, although it is not improvisation, because neutral red is still used, because if we have to do a plaque purification, if we have to take a colony out of the mixed viruses, neutral red is still used, because the viruses are still alive when neutral red staining is done. Whereas the crystal violet staining, because there is a fixative agent like acetone, methanol, ethanol. So the viruses and everything gets fixed, and then wherever the cell lysis has happened, we can clearly count the plaques. Then the most advanced or the recent development is the immunostaining, because counting them is very difficult, like in these two cases. Sometimes the pinpoint plugs get missed, but most importantly, if we are working on a tetravalent vaccine, for example, dengue, if we are working, it's a tetravalent live attenuated vaccine, although it is not in the market, but in several companies it is under development. So if it is a tetravalent vaccine, we have to show as per the COA that we have all these 4 serotypes in our formulation. But then how do we show all 4 serotypes? Real-time PCR is of course one method, but immunostaining is another good method where we can have replicates of dilutions of plate, and then each plate is stained with specific monoclonal antibodies, so we can identify and titrate each of the virus in a mix of tetravalent vaccine. Similarly, the TCID50 or the endpoint 50 assay, this is another method of quantifying a virus. There are some viruses which don't form plaques, but they form some cytopathic localized formations on the cells. So this method is very good for such kind of viruses. So now, while till now we have just talked about how viruses are quantified and what all happened in that field, but the concept of virus neutralization, why virus neutralization has to be done. So that has to be done because of this term, the correlates of protection. Correlates of protection. So in 1796, when Edward Jenner did his experiments to prove or develop a vaccine, that time survival was the only indication. A person surviving with smallpox infection and he is not dying or not showing critical symptoms was the only evidence to demonstrate that it is working. Because as I told, there was no concept of even viruses existing those days. So there was some notional thing that something is happening with this infection and people are dying. So this famous experiment, I can just briefly take you, like a lady with cowpox infection, those scabs were like introduced into a 8-year-old boy, James Phipps, and then he got immunity against cowpox and also smallpox. Then another lady infected with the smallpox, the infected pock marks, like all the pus material, that was introduced in the same guy, James Phipps, and then he survived. He didn't show. So this was the first, and then Jenner reported 20-30 more cases, and then it was accepted that okay, this is working. But those were the days. But now we have to show much more in vitro data, which is known as correlates of protection. And that correlates of protection has to directly correlate with the immunity. So what are correlates of protection and why are they required? They are required to evaluate the consistency of vaccine production, because when we are manufacturing vaccine, each lot of vaccine should have the same consistent antigenic profile, immunogenic profile of that antigen. So for example, if we get a, for example, say 100 milli-IU antibody response of one batch, all the batches should give in and around 10 plus minus % of that kind of. So that shows that our vaccine is consistently manufactured. Second is to check the susceptibilities of individuals and populations after vaccination. So Dr. Manoj this afternoon also told that why we have to study in different populations, because we should not expect the same immune response in each population. So we have to establish customized specifications of immune responses in each population, if that is the case. Then it is also used to assess, means the correlates of protection are also used to assess the next generation or combination vaccines when a prior generation vaccine is licensed. So to define correlate, it is an immune response that is responsible for and statistically interrelated with protection. And there is a surrogate. This is also a term which is frequently used in immunogenicity. A surrogate is an immune response that substitutes for the true immunogenic correlate of protection, which may be unknown or not easily measurable. That doesn't mean that surrogate cannot be a indicator of correlation. Like over the time with lots of data, we have to establish a surrogate which almost every time correlates with the real correlate. Then the regulator also accepts the surrogate values also. So the correlates of protection and establishment of surrogates. So of course, virus neutralization is a gold standard. So if we are working on any novel vaccine or any zoonotic new emerging viruses, so there are no established correlates of protection. So in that case, we have to do virus neutralization assays. Like in COVID, everyone has to do had to do the neutralization assays because there were no direct markers of protection. But then specific antibody levels have been proven to strongly correlate with defined neutralization titers. So this is what is the surrogate, specific antibody level. So I'll show you some examples. So let us see, like in case of varicella, earlier days, whoever has manufactured the varicella vaccine, definitely they would have done the neutralization assay. But now, over the time, it has been established that any antibody level against the specific glycoprotein, if it is equal to or higher than 5 ELISA units, like it can be accepted as protective. Similarly, for IPV, like some titers have been defined, and for hepatitis A, of course, NIBSC standards are available, and 20 milli-IU is the cutoff to show that this vaccine is protecting against hepatitis A infection. So we need not do neutralization. But in some cases, like for Japanese encephalitis, PRNT is the standard. There's no surrogate established as of now. So when we talk about viral vaccines, there are like two major types of vaccines. One is kind of JEV, measles, polio, where we can do neutralization of the virus. We are talking about the human sera for neutralization. Then there is another set of vaccine like the influenza vaccine where the neutralization doesn't work. The hemagglutinin inhibition works, where we have to show the HI value of neutralization. But we will talk more about the first segment. So continuing with the plaque assay, we can do the neutralization. It is like very simple. We simply dilute, we can make a dilution of the sera, which has to be tested, and then we can neutralize a fixed quantity of virus. And once the neutralization is done with some incubation at say 1 hour or 90 minutes, we can introduce it onto healthy monolayer of specific cells, and then we can incubate them further after the overlay and all. But this method is cumbersome because say, if we are using a 6-well plate and if we have 6 dilutions, one plate is like one set, not even in duplicate. So if we want to have triplicates, we need to have 3 plates for one sample. Similarly, if we do the TCID based assay, so before adding the virus, like we neutralize the virus and then we do the TCID assay, that we can also do in 384 well plates. So the conventional method is like we have to look into each and every well through microscope, which is very cumbersome. So that is one challenge for doing the TCID assay neutralization using the microscopy method. But then there have been some improvisations. So the immunoplaque assay, which I was talking some time back. So this is like one method where we can cut down the time of microscopy and like plaque counting, and this immunostaining is more specific because it is a monoclonal antibody based, or we can also use polyclonal in several cases. And then when we talk about microneutralization, the improvisations are like there now machine readable formats are available. I was talking to someone from Beckman today, and they said they have fully automated systems which can be used to do TCID assays and also for neutralization assays. So what are the challenges? Of course, to make it quicker, the highly pathogenic, or if you are working on some emerging viruses, zoonotic viruses, we need a BSL-3 or BSL-4 facility, which is like for manufacturing itself is like having such facility is difficult. Again, to have another such facility for QC testing is even more challenging. Then the CROs also, like, because these are not everyday kind of contracts to get. So to maintain that kind of facility also is very not very cost effective for the CROs. So then what do we do? Like, what is the way out? So the way out is the most important or promising amongst all these developments, recent advances, is the pseudovirus assay. Now, what is a pseudovirus assay? As the name suggests, a pseudovirus is a virus expressing surface antigens of the virus of interest. A pseudovirus mimics the natural virus and helps assessing the neutralization potential of antibodies. Now, a pseudovirus can be a capsid completely made up of surface antigens, like in case of HPV or polyoma virus, because like the pseudoviruses of these viruses are completely made up of the same proteins. In case of HPV, it is all L1 and L2 proteins. And pseudo-typed virus is another type of pseudovirus where the surface antigens of one virus are expressed onto another virus. Say, for example, for SARS-CoV-2, we have used lentivirus systems to make to generate pseudo-typed virus. So, just to explain how a pseudovirus is designed, so we have the cells. Normally, the HEK 293 T cells are used, and for example, I'm talking about the HPV pseudoviruses. So, we take the L1 and L2 expressing plasmids, and then we have a reporter gene, and once they are transfected into the cell line, we get matured particles which are having the gene, the reporter gene trapped inside or encapsulated inside. So, these are the pseudoviruses. Now, they are exactly mimicking the original wild type virus. So, this does not require any BSL-3 or BSL-4. It can be happily done in a BSL-2 facility, and it exactly mimics the wild type virus activity, like the tropism and all that. And talking about pseudo-typed virus, I have taken an example of SARS-CoV-2, and I have taken an example like there are like lentiviruses and other oncoretroviruses. In lentiviruses, HIV-1 is the most widely used lentivirus, and then there are several generations. Now, 4th generation lentiviruses are being used. So, here we have a packaging mix where we have all the genes coding for the enzymes and all to make the lentivirus. Then we have the transfer vector where we have the reporter gene and then the spike protein. In case of SARS-CoV, we will have the spike gene coding for the spike. Then once they are transfected, we have these pseudo-typed viruses coming out where the spike proteins are expressed onto the lentivirus frame and the reporter gene is inside. But there's one more requirement to pseudo-typed viruses. In case of COVID, because everyone, like we know that the COVID, the virus, the coronavirus needs the ACE2 receptors to enter into the cell. So, then we need a cell line which expresses the ACE2 receptors. So, there are like other like set of experiments through which we can transform the HEK293 cells to express the ACE2 receptors, and then using those two ACE2 receptor expressing HEK cells, we can do the neutralization assay. So, we can incubate the pseudo-typed viruses with a range of diluted sera and then infect them. So, whichever pseudo-typed viruses are not neutralized, they will have specific receptor based entry into the cell, exactly mimicking how a wild type or natural infection happens, and then the reporter gene will start emitting the signal. And again, all these are machine readable formats, which is like we can have a high throughput testing. So, and these are the reporter genes which are used, like the sources of reporter genes. The most popular is the nanolook, which is very small in size and which gives very good readout. So, I tried to rush and tried to finish in time. So, in summary, pseudoviruses, the pseudovirus neutralization assays mimic the natural wild type infections. These novel methods provide alternatives to the gold standard PRNT assay, and the regulatory authorities have accepted, or they accept these methods for highly pathogenic and emerging viruses, because now with somehow like thanks to the COVID outbreak, now the regulatory authorities are more accepting these requests, and they are also understanding the challenges of unnecessarily allowing the PRNT assays using wild type viruses, because again it will lead to some other environmental exposures. Yeah, I end my presentation. Any easy questions for me? Okay, thank you.
Thank you, Dr. Vinod. And now we move on to our next presentation. Our next presentation will be from Dr. Rajendra Burki, Associate VP R&D Biological E Limited. Dr. Rajendra will speak on R&D analytics strategies that can help speed up vaccine lot release without compromising quality. Dr. Rajendra.
Meanwhile, you can display. Thank the organizers for inviting me to talk about this interesting topic on analytical strategies for different vaccine candidates. Okay. So the opinions expressed in this presentation and in the following slides are solely of the presenter, not representing the biologically limited in any manner. It's not moving. Sure, sure. No problem, that's okay. Sure, sure. So Vinod was asking for